There are three ways in which water is going to diffuse. Remember, diffusion of water is known as osmosis. We have this first picture representing a hypotonic solution. You can think hypo like hippo. Hippos are big, so the cell gets big. In order for the cell to get big, the water is going to diffuse into the cell. The middle diagram is showing an isotonic, which remains at equilibrium. For every water molecule that moves into the cell, a water molecule will move out of the cell. And then the third diagram is hypertonic, where the water molecules are moving out of the cell, and since water is leaving the cell is going to shrink. Let's look at some examples of osmosis, which is the diffusion of water across the cell membrane. In looking at our first one, we have hypertonic. In a hypertonic solution, notice that there's, let's get my pen. Notice we have 95% water in the cell and 85% water outside the cell. Well, we're trying to equalize that water amount, so water's going to have to leave the cell in order to lower this concentration and raise this concentration, because we're trying to get them to be the same. This would also lower the salt concentration. So water has to leave the cell, making the cell smaller. That's what's known as a hypertonic solution. In this second example, we have an isotonic solution. Notice my water content, 90% water in the cell, 90% water outside the cell. Since my water amount is the same inside and outside of the cell, I'm not going to have any net movement. I can have movement, but if 5 water molecules move out, 5 water molecules would move in. This is what's known as an equilibrium, where the amount of water that diffuses into the cell is going to be the same as the amount that diffuses out of the cell. And since the amounts are the same, they say that's no net movement, meaning no movement of particles moving to one side and staying there. Our third example is a hypotonic. Remember, hypo like hippo gets big. I have 80% water in the cell, 90% water outside the cell. So I'm trying to make the water amounts diffuse to the same. Water's going to have to go into the cell to lower the outside content and raise the inside water content. Water goes in, that makes the cell big. That's hypotonic. This is showing you 3 red blood cells. This first one is representing a normal red blood cell. They kind of look like a little disc shape. That's what cells should look like. In this case, that's representing isotonic. The second diagram is showing a cell that has gotten plumped up. It's gotten bigger. Water has moved into the cell. It's gotten bigger like a hippo, so it's hypotonic. And then the third one is showing that a cell is beginning to shrivel. Since the cell is getting smaller, water is moving out of the cell. That's hypertonic. This again is just showing you the red blood cells where we have the bottom one down here, isotonic. Water moves in and out at the same rate. We have hypertonic, where the water is leaving the cell. As a result, the cell is starting to shrink. And then hypotonic, water's moving into the cell, making the cell plump. Okay, taking a look at cells placed in a different environment, meaning different solutions. This first one we're looking at are red blood cells. The red blood cells are representing animal cells, and in the first one you'll notice that the cell has begun to swell, indicating that water is moving into our cell. The reason water would move into the cell is because there's a higher concentration of water or higher water potential outside the cell. So the water moves from high potential or high concentration to lower potential inside the cell. That represents that the cell is in a hypotonic solution. In the second one, the cell is beginning to shrink, indicating that water is leaving the cell. It's leaving the cell because it's going to, again, have water move from a high potential. There was a higher water potential in the cell. It's going to a lower water potential outside the cell. That means that the cell has been placed in a hypertonic solution. The last one represents that nice disc shape for a red blood cell. Therefore, you know that you're looking at an isotonic solution because the water is moving in and out at the same ratio. On the bottom ones, we're looking at plant cells. Plant cells have a cell wall, and that cell wall acts as a protection, and it helps maintain the shape of the cell. So as this one, you'll notice the cytoplasm is beginning to shrink. This is indicating that the water is leaving the cell. As it's leaving the cell, the cytoplasm begins to shrink, but you'll notice again your cell wall is going to maintain the structure of the cell. Therefore, this cell has been placed again in a hypertonic solution. This is what's known, if you'll notice on your guided notes, this is what's known as plasmolysis. Because of the cell wall, again, the cell shape stays for plant cells. As a result, the cytoplasm begins to shrink, and that's what we're noticing here. And again, that's called plasmolysis. On the middle one, we have that nice rectangular shape, so we know that water is leaving the cell. Okay, my pen's not working today, so I'm not going to worry about that. But water's going in and out at the same ratio. Therefore, we know that the cell is in an isotonic solution. On this last one, we notice that the cell is beginning to plump up. As a result, that indicates that the water is going to move into the cell. Water again goes from a high potential to a low potential, so there's a higher water potential outside the cell, lower water potential in the cell, indicating that that cell has been placed in a hypotonic solution. Another thing I'd like to mention for the animal cells, remember, don't have a cell wall. So as water would continue to move into an animal cell, such as the red blood cell, it can only take in so much water. Eventually, if it took in too much, it would be a dangerous situation. It would actually burst. In a plant cell, again, you have the cell wall that's going to help prevent that. The cell wall will allow so much water to enter. When a plant cell takes in maximum amount of water, that's what's known as a turgor pressure. So we've talked about diffusion, where particles are going to move across the cell membrane from a high concentration to a low concentration. We also have a passive transport that's facilitated diffusion. Facilitated meaning that there is going to be something to help in this process of still going from high concentration to a low concentration. Notice there's more particles, more purple particles at the beginning over here, and they're moving to a low concentration across the membrane, but they're moving through a protein channel. You'll notice this one over here does not have a protein channel. The facilitated diffusion has helped getting particles across the cell membrane. This is representing a cell membrane where we have facilitated diffusion. The molecules are going to move through the pores or the protein channel. This is representing your proteins. This is the channel in between them. And so this is allowing to make basically a little opening or a pore. Those protein channels make an opening for the particles again to go from high concentrated to low concentration. Facilitated diffusion. Facilitated diffusion is a passive transport. It does not require energy. You're still going from a high concentration to a low concentration. And looking at your diagram, this green structure is representing your protein. And then here's that channel that allows for the particles to move through. Examples of particles that need facilitated diffusion: glucose or amino acids moving from blood into the cell. Types of transport proteins. You have channel proteins that are embedded in the cell membrane and have a pore or an opening that materials can pass through. And then carrier proteins can actually change their shape to move materials from one side of the membrane to the other. Okay, looking at the fluid mosaic model or the cell membrane, you see some of the different proteins. These blue structures are proteins that are throughout the cell membrane. You have integral proteins. Integral proteins actually go all the way through. You'll notice that it starts here and you can trace it all the way through to the other side. This one, if you're looking up at the top here, you can see it through the fatty acids and comes out. So they actually can go from one side to the other. Sugars and amino acids use the integral proteins. You also have peripheral proteins. Peripheral proteins are found on the outside of the cell membrane. And then if you attach a carbohydrate, this is representing a carbohydrate. This is your protein. When you attach a carbohydrate to a protein, it's known as glycoprotein. The glyco is your carbohydrate, and then it's attached to a protein. You could also find carbohydrates attached onto lipid molecules. And in that case, they would be called glycolipid, where this is the glyco portion is the carbohydrate, and then the lipid, which is below. We can't actually see it in this diagram. And remember, this is fluid. Those proteins are able to move throughout the mosaic. Just another diagram of looking at different proteins. Your channel proteins, they form a channel for substances to pass through, kind of like a little bridgeway, where they're able to go from high concentration to low concentration. Then we have carrier proteins. Carrier proteins actually can change their shape. So you'll notice in this diagram where the protein is open to the outside of the cell, it takes in a particle, and then it's going to close back up as that particle moves to the inside of the cell. So carrier proteins can actually change their shapes. Proteins are important for membrane function. We have 4 different types of proteins: our structural, recognition, communication, and transport, all of which are going to help with the functions of the cell membrane, moving particles across the membrane. Let's look at active transport. Cells move molecules against the concentration gradient. In order to go against the concentration gradient, particles are going to move from low concentration to high concentration. That's not the normal flow, so it's against the concentration, low concentration to high concentration. In order for that to occur, active transport is going to require a protein pump to do this. And that protein pump is going to need the help of energy, chemical energy, in the form of ATP. in order to move those substances from a low concentration to a high concentration. Active transport is another form of moving particles across the membrane. However, in active transport, you're going to need the help of ATP. ATP is supplying the energy that is needed. And in ATP, you're going from low concentration to high concentration. This is against the concentration gradient. You'll notice when you look at the pictures here, there's more green particles on this side of the cell. So the energy is needed to move from low concentration to a high concentration against the concentration gradient. Same thing in this second diagram. There's more little particles on this side. That's your high concentration. Energy is needed to take low concentration to a high concentration. An example of an active transport is using the sodium potassium pump. Again, with active transport, you need energy. That energy comes in the form of ATP, and it's moving against the concentration gradient. In other words, going from a low concentration to a high concentration. Proteins can transport materials against a concentration gradient. Proteins called pumps move materials against that concentration gradient. Active transport drives molecules across the membrane from a low concentration area to a high concentrated area. The pump, remember we've talked about one that was that sodium potassium pump. Okay, the sodium potassium pump, again, this is using energy, a form of active transport. The sodium potassium pump is going to take 3 sodium ions, and you'll notice those 3 sodium ions actually fit inside this protein pump. Also with the form of energy, where you have ATP. ATP is going to release one of those phosphates. The phosphate attaches to the pump. It's allowing this pump to change its shape, moving those sodium ions from a region of low concentration to high concentration. When the sodium ions are released, the potassium ions, you have 3 sodium ions released, 2 potassium ions are then going to go into the pump, that protein pump, allowing them to be released on the other side along with that phosphate group. So the sodium potassium pump uses energy directly from the breaking down of the ATP releasing the phosphate group. It pumps 3 sodium ions out of the cell for every 2 potassium ions it pumps back into the cell. Taking a look at endocytosis and exocytosis, sometimes a cell may need to use energy to move a large substance or large amounts of a substance. And to do that, they would move those substances in vesicles. When they're transported in a vesicle, it allows that substance to enter or exit the cell without having to cross the cell membrane. Endocytosis, think in, things moving into the cell. This is a process of taking either liquids or fairly large molecules into a cell by engulfing them in a membrane. And if you look at the diagram at the bottom here, you can see where we have a particle and it is getting engulfed by the membrane. That membrane is going to wrap around, it fuses together to close, and then it's going to pinch off and form a vesicle moving into the cell where it needs to go. There are 2 types of endocytosis. The first one is phagocytosis, which involves engulfing large particles. It's kind of like a cell eats a cell. This is important in your immune system. An example would be white blood cells killing foreign molecules or bacteria. Then the second one is pinocytosis, which is also going to involve the movement of particles into the cell, but now we're looking specifically at the movement of liquids. Then we have exocytosis. Think exo like exiting, leaving the cell. Exocytosis is the release of substances out of the cell by a fusion of a vesicle with a membrane. So again, you have a vesicle that has substances in it. It's going to fuse to the membrane, and then once it fuses to the membrane, it'll open up and release those substances out of the cell.